this video, we will discuss about the invasion and metastasis of pancreatic cancer in Galectin-1 overexpressed pancreatic stellate cells. We will discuss the in vivo results that they have presented in the paper, which uh, only validates their in vitro findings that in the Galectin-1 overexpressed pancreatic stellate cells, the pancreatic cancer cells will have more EMT characteristics. So first they validate the whether these EMT characteristics also observed in the in vivo results or not. So they take the orthopathic models, orthopathic injected mouse models, and did the H&E staining. And what they are able to see is that E-cadherin levels are high. E-cadherin's levels are high in the case of this Galectin-1 is been knocked down, whereas and we can see that Galectin-1 signals are low, and when the Galectin-1 signals are not affected, or when they use wild-type pancreatic stellate cells, they can see the E-cadherin levels are at mediocre levels, whereas when the overexpressed Galectin-1 is used, it can be visible from this H&E staining also. We can see a low level of E-cadherins. However, the markers for the mesenchymal, that is Vimentin, they are able to see that Vimentin levels are less in the case of Galectin-1 knockdown, and moderate in the case of wild-type Galectin-1, and are overexpressed or enhanced in the case of overexpressed Galectin-1. And these things are quantified in this panel J and K. So in the panel J, we can see that the validations of the pancreatic stellate cells, that is whether they have overexpressed Galectin-1 or Galectin-1 has been knocked down. We can see that the quantification shows the same results that we saw in the H&E staining. And similarly, they also quantified E-cadherins and Vimentin signals, and we can see that E-cadherins signals are high in the case where Galectin-1 has been knocked down, whereas Vimentin signals are high in the case where Galectin-1 is overexpressed. So this indicates that these pancreatic stellate cells that have this overexpressed Galectin-1 will cause EMT changes. And in order to further validate their EMT changes, they check the invasion and metastasis of those pancreatic cancer cells in vivo. And what they are able to see that in the case of this liver, so these results are for the case where we will discuss some of these figures. That is for the panel F, it shows that the pancreatic cancer cells are towards this invades towards the wall of the stomach, and some of the metastases are also seen in the case of liver. And this panel G shows that epithelial levels are low, and panel H shows the Vimentin levels are high. So these results are mainly for the conditions where pancreatic cancer cells are overexpressed. Pancreatic cancer cells are injected along with the pancreatic stellate cells that have Galectin-1s overexpressed. And we can see that metastasis is clearly evident in the panel I also. And this panel J shows the control conditions in which there is no mice is not infected with the pancreas, and panel K shows the healthy pancreas, and panel L shows the healthy pancreas with a star, whereas panel A shows the pancreas that are affected pancreatic cells that are the tumors of pancreas, where pancreatic cells have cancers properties. And in the panel B, we can see that this shows the tumors in the pancreas, and panel C shows the epithelial levels that goes down, and panel D shows the Vimentin levels that goes up. So this all indicates that it's this Galectin-1s somehow interact with these pancreatic cancer cells and trigger these EMT changes. However, it is not yet known that how these pancreatic stellate cells move along with the cancer cells, and this needs further investigations. That is what they have said in the paper. So I hope you understood how they validate their hypothesis that galectin-1's overexpressed pancreatic stellate cells helps in vision metastasis of the pancreatic cancer cells, or PDAC. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.